Really pleased to be here in support of the foundation, and also to try to give you, I was going to say a few words of wisdom, but I'm not sure I'm quite there yet, actually. When it comes to understanding the brain, what I don't know about the brain far outweighs what I do know, and that's because it's an extremely complex little creature to kind of understand. But I have spent my whole life trying to understand it. Well, it seems like my whole life, and brain plasticity has been the kind of the central theme or focus of all the work that I've been undertaking. And as my work has evolved from understanding basically kind of how memory works and how the brain kind of normally functions, it has then segued into trying to understand what it means and how those things change when the brain isn't functioning quite so well, and whether the capabilities of the brain can still kind of the brain has its own ability to kind of try to work on itself and help itself through its brain plasticity. So what is the brain? It looks like this from a top view. It's not, I suppose, really normally shiny like that, but anyway, you can see that's quite a large structure. It looks convoluted and complex from the top view, but actually if you then cut a brain halfway down through the middle, you cut it down like this, then you'll see that it's actually even more complex inside. So there's lots of different regions. They all communicate and talk to each other in different ways. And some interesting facts. These are not fake news. This is real news. It weighs only about 1.4 kilograms, plus or minus. So it's only about 2% of your total body weight, okay? But in that 2%, there's something like 100 billion nerve cells. And that's only one of the types of cells in the brain. It's got as many or more of other types of cells in the brain as well. And yet that's 2% is using up 20% of the cardiac output, right? It sucks that blood in. It needs all that continuous blood supply, the oxygen and the other glucose and other things coming in to keep it all going. And it comes in through this, which is the representation of the blood supply. You can see that the blood supply to the brain is immense. It's complex. It has to get everywhere. No nerve cell can be more than about 20 or 30 micrometers from a vessel, so that it can get the supply coming in that it needs. So there's 22 meters of blood vessels going through the brain. And so that's about 2.5% of the total brain volume. And this is critical, of course, because if there's anything that impairs the blood supply of the brain, it's going to impair brain functioning. So keeping a good blood supply is very important, among many other things. So the brain, I like to call it plastic fantastic. This left hand is something I just pulled, an image I just pulled off the internet. It's actually a book, which I've never read, and I'm not advocating that you do read the book, but I thought it was a nice image. So anyway, it's brain plasticity is what I'm going to talk about, and there. It's got buckets full of plasticity, which I'm about to explain. This is a plastic brain. But that's not what I'm talking about. I'm not talking about plastics. I'm talking about plasticity. And I like to go back in history for these talks, because I actually work in the psychology department at the University of Otago, and my office is in the William James building. And William James, who, as far as I know, has no connection whatsoever to New Zealand or Otago, but in our wisdom, we named our nice new building the William James Building, because William James is like the father of modern psychology. And he wrote extensively, but he wrote a very kind of groundbreaking book called The Principles of Psychology back in 1890. So what's that, 130 years ago? And he used the word plasticity back then, I think, for the first time with respect to brain function. And I have this great quote. It's a little bit kind of ancient, kind of archaic way of writing and speaking, but I think it's fantastic. Plasticity then, in the wide sense of the word, means the possession of a structure weak enough to yield to an influence, but strong enough not to yield all at once. And basically that's what we talk about when we talk about plastics, right? Particularly soft plastics. You can move them, you can manipulate them, and they don't break. I could probably twist this bit of plastic, right, and it doesn't break. And it retains its shape more or less. Of course, there are hard plastics, and you twist them and they break. So, I mean, there are different kinds of plastics. But this definition is really relevant to how the brain works. Okay? It has the possession of a structure weak enough to yield to an influence. The brain can be influenced, and it can change as a result. So then, organic matter, especially nervous tissue, the brain, seems endowed with a very extraordinary degree of plasticity of this sort. So that we may, without hesitation, lay down our first proposition, the following: that the phenomenon of habit, and by that he means like memory and learned behaviors and motor skills and so forth, in living beings, are due to plasticity of the organic materials of which their bodies are composed. And by this he means really the brain and the nervous system. So the idea that the brain is plastic, that it's not fixed, but it changes, and this is how we learn and do all the things that we do as human beings, and as animals do as well, is based on this plasticity. So the first thing I want to do is just talk about different kinds of plasticity, because, as I mentioned, the brain has buckets of it, so there's got to be more than one kind. So first of all, think about development of the brain. So the neural tissue starts as kind of just about microscopic level, and then during development it grows and grows and grows, and the brain gets bigger and bigger and bigger. And in this diagram, you can see just the growth of the preterm infant, baby, 28 weeks going up to 38 weeks. So they can see there's a massive growth of the brain. Okay, so it's adding brain cells for a start, but those brain cells are also expanding their reach and their connections, and so they are themselves getting bigger as they're added in and developing over time. And you can see that the brain also gets more convoluted. This is the outside of the brain, this part here, and the kind of the top part of the brain, which has all these kind of indentations and gyri and fissures and so forth, because actually the brain wants to be bigger than the skull is letting it, right? If the skull was bigger, the brain would just get bigger and you wouldn't have all these indentations. But because the skull is holding it in, as the brain during evolution has evolved, it's had to create increased surface area by creating convolutions. And so that happens during development as well. Okay. And so it's becoming bigger, more complex, and so the brain is wiring up. The cells are beginning to connect to each other. Okay. To a large extent, this is genetically controlled, but it's also controlled by the environment, even in terms of how the cells are actually operating based on kind of the influences that are coming through the mother at this stage. So it's already plastic before birth. And as it's wiring up, you have kind of wonderful sets of connections going across between the different parts of the brain, across the two hemispheres, up and down the length of the hemispheres, up and down from the top to the lower structures, and then back up again. And so this image here is actually what can be created from magnetic resonance imaging, MRI scanning using particular protocols. People can kind of use computer calculations from the data to draw these images, create these images showing the connectivity of different parts of the brain. And the interesting thing is, no two brains are the same. Okay, so if this is all genetic, of course, everyone's genes are different too. But even identical twins would have somewhat different connectivity, right? Because the connectivity develops in response to the experiences that the person is going through. Okay. So no two brains are the same. Thank goodness for that. Because I'm pretty sure you wouldn't want my one. Now, the other thing is, and this is kind of following on that theme, that the connectivity, as I was saying, it changes during development. And I kind of think of it like kind of from a kind of a personal level. So as you go through school, for example, or daily life, you meet lots of people, right? And you meet, as you go through the grades, for example, in school, you meet more and more and more. But you don't kind of remain connected with them all over time. There are only some that become special for you, right? And that you maintain relationships perhaps for much, much longer. So in a way, the brain's the same. So when it's trying to hook up, this little diagram of visual cortex development. When we're hooking up the nerves from the eye, back from the back of the retina, going into the lower parts of the brain, into the higher parts of the brain of what we would call visual cortex, when it first hooks up, you can see that it's a very, what we might call exuberant projections. Everyone wants to talk to everyone else. It's like the kids in, first grade, year one. But as the person or the animal has more and more visual experience, then it turns out that the left eye really only ends up talking to one part of the brain structure and one part of the cortex, where it first comes in. Eventually, of course, it mixes. But then the right eye talks to other parts. So this initial exuberant connectivity, based on the experience, the visual experience that a person has, starts to separate out in the way that it's meant to be. But you can think about this. If we knew exactly how it was meant to be, it could just be all genetically determined, right? They could just wire up the right way to begin with. What's the point of having all this exuberant connections and then kind of retracting them and formulating the right connections? Well, it's because it's leaving itself open to forming the kinds of connections that are relevant to the environment and to the visual experience that the brain is having. Okay? So it's leaving itself open for change. And then it will change in response to the visual experiences that the organism is having. So the brain is built to wire up in certain ways, but then to adapt its wiring based on experience. So it's a very particular kind of plasticity that goes on in development, but many of the actual mechanisms that mediate these changes stay online during adulthood and can contribute to other kinds of brain change as well. So what other kinds of brain change are there? Well, here's one, which is a brain section viewed at very high power through a microscope, looking at a particular part of the brain called the hippocampus. You may have heard of the hippocampus, but if not, it's a brain structure that's important for learning certain kinds of information. So it's my favorite structure, I must say. Very beautiful structure. It is. And what is labeled here, so in blue are the cell bodies of all the main cells, the nerve cells in the hippocampus. But in red are cells that have been recently born in an adult animal. In this case, it's, I think, a mouse. All right? So there are actually stem cells in the brain still going right through life. Not just rodents, humans as well. Okay? So there's a constant supply of new cells in this region anyway throughout life. And like in a human, it would be several thousand a day, right? Just keeps pumping out these cells. Unfortunately, the rate sort of goes down with aging, sadly, but it's still going. Now, this is not for brain repair. This is an important aspect of the way adding new elements to the circuitry is quite an important part of the way in which the hippocampus processes information. And I don't need to go into the details of, well, mostly because we don't actually know why exactly, but there's lots of theories. But it is very critical. And if you impair the ability to produce these new nerve cells in adulthood, you can impair learning and memory function. And actually, you can also get other changes. For example, it can lead to depression and so forth. So it's multifunctional. So this is a kind of plasticity where we keep adding elements to the network. And so we call this neurogenesis, or generating new nerve cells. So I don't know if to say unfortunately. There are stem cells in various parts of the brain, but there's only a couple parts where they're really active. But the potential is there for some of these stem cells to come online and become more active when there's brain damage. Now, so far, we don't really know that they can actually do much in the way of actual repair. But there's a lot of people out there who have hope that we might be able to harness this kind of plasticity for brain repair when there's brain, nerve cells are lost through disease or damage. But at the moment, it serve normal functions anyway, so that's quite important. The thing that I've studied mostly is synaptic plasticity. So of course you're probably aware that nerve cells contact each other at what are called synaptic junctions. So one cell, this is a very interesting artistic rendition, but one cell has a little process that comes in and butts up against another cell's process, and there's this synaptic junction here where chemical neurotransmitter is released and jumps across this very tiny junction and activates protein receptors on the other side, and the next cell becomes activated. So if we look at a real bit of nerve cell process, which we call a dendrite, this is the receiving cell, how it looks like, you can see that there's all these little what we call spines, and these red bits are where the incoming projections from the incoming cells actually make contact and where the synaptic transmission actually occurs. Now, if you look at the variety of the size and shape, the length, the width of these different points of synaptic contact, you can see that there's a huge variation, almost 100-fold size and contact points. So, and this is not genetically coded. This has to have happened as a result of experience. So as a result of experience, some connections grow and get bigger. Some will get smaller and weaker. Okay, and I guess I'm just going to show you, before I get much farther, I'm going to show you where another picture where now we have not just the receiving cells, spines and contact points, but now we have the green incoming fibers from other cells, and you can see that they make contacts at different points along this dendritic process. So the point here is, though, that there's very strong evidence. There's a lot I don't know, but there is one thing I do know. There's a lot of strong evidence that memory involves changes at these synaptic contacts. Okay? And the idea is that you want the cells that have been activated together during an experience to become more strongly connected. So when you go to remember that experience, it's easier to reactivate that network that was activated in the first place. Right? If they're more strongly connected, then as you start to kind of re-experience something or remember it, then the whole network fires because they're so strongly connected. And then, of course, but this is also the ones that don't fire together very often become more weakly connected. That's kind of like that pruning of the exuberant connections I talked about during development, but on a more subtle level in the adult as a part of learning and memory. And you can see that, gosh, there are a lot of synapses. For you mathematicians, that's 10 to the 15th. But you can think of it as a billion trillion of these little contacts in a human. And they're modifiable, they're plastic as a result of experience, and they can more or less be changed one by one. So as a result of experience, you can modify some here and some there and some over here, and then for another experience of memory, it's different ones. And so the storage capacity, the processing power, is huge. Computers have yet to kind of reach this level of processing. No doubt one day they will, and that they might not be so far away. But what nature has been able to achieve is pretty amazing, awe-inspiring. Okay, so what happens when these cells that are active together at the same time and their synapse has been firing away, and so then basically some molecular mechanisms are activated in the cells, and this is a strengthening of these contacts. And so this is just a little diagrammatic representation of what we call now long-term potentiation of synaptic strength. And in the field we just refer to it as LTP, long-term potentiation. And what this means is that the result of repeatedly activating these synapses, as you have the experience and then as you rehearse it, maybe during sleep, or as you think about it, you continue to rehearse those same neural circuits that are involved in the memory. And I'm very sure that tonight, as you sleep, you'll be rehearsing my talk and strengthening those synapses about brain plasticity. So there's a growth of the contact zone, and there's more transmitter release from the incoming fiber, and there's more of the receptors that the transmitters contact in the post-synaptic or in the receiving cell spine. Okay, and so it's functionally more effective, and you can see morphologically it grows as well. And in the lab, we can when we studying this, we can measure this using experiments where we can record the electrical activity of the incoming and the incoming fibers and the receiving cells. And we record how strongly this response is, because this is really electrical response that the cells are using. The brain works on electricity. And so we can measure these kind of funky waveforms on an oscilloscope or with a computer. And we can see that, so in a standard experiment, we have, just the occasional synaptic transmission that bumps along, and there's always variability because it's a biological system, but you have some kind of baseline. And then at this arrow, we kind of pretend that it's learning something. So we give very high frequency activation, but very briefly, to this circuit, and because this is exactly what happens in the brain. When you're learning something, the cells that are processing that information start firing like mad for very brief periods of time. And that's all it takes. And as soon as that happens, you can see that now the size of the response is bigger. And when we measure it, then you can see now it's a much bigger response than we had before. So this is the experimental way of studying synaptic plasticity, by kind of artificially giving the network a memory, by giving the kind of neural activity as if it were actually having an experience. And you can do these kinds of experiments actually by having animals actually have an experience, and you get very similar things if you record in the right synapses. Now, if you have, a billion trillion synapses, finding the right ones to record from when the animals have an experience can be challenging. But yeah, we find ways through that. Now, plasticity also occurs in another domain, and that is in response to disease or injury. And, people will have strokes from time to time. And there's various, risk factors around stroke, but sometimes it just, they just happen for unknown reasons. So here's MRI scans, normal brain, and a brain that has had a stroke here, so in this white part. And you may well know people who have had stroke. And sometimes recovery from stroke is quite remarkable. Okay, sometimes it's big enough that there's just no full recovery, and that's not uncommon, because if you actually have a lesion, if this whole part of the brain has died off, that's pretty difficult to recover from. But nonetheless, other parts of the brain can begin to take over some of those functions that have been lost by further experience and rewiring. And so rehabilitation is really basically trying to undertake this process to help people have the right kinds of experiences so that the right kinds of rewiring can occur, so that critical functions can be restored. And it's not just about kind of motor function, of course, but also various cognitive functions and so forth that might be affected, language and so forth. And it may never be completely right, because you can imagine if you took out a piston of the engine, no matter how hard the other pistons work, it's hard to get the full power right out of your engine. But nonetheless, you could do quite well if you had enough power. So that is also an important thing. And many of the same processes that we are involved in normal learning and memory are brought back into play to help with rehabilitation, because it's all about the experience the person is having. Now, I'm also going to suggest that even things like counseling, okay, or clinical psychologists or psychiatrists or whatever they're doing and helping people with issues they're having, you might think of this as like psychological. But you can't have a change in behavior without some kind of change in the brain. So people are relearning and reworking through their circuits so that if they're having memories that are too strong, like post-traumatic stress disorder, if they have addictions, or if they're depressed, because the brain's working in an inappropriate way, then through the right kinds of experience, contact with other people, and working through strategies, you can kind of work through how the brain is going to behave. Okay? So this involves change in the brain. There's no doubt about that. It's not just psychological, because there is no such thing as just psychological. Everything that's psychological is built on what the brain does. Okay? So if the behavior is changing, the way of thinking is changing, the brain is changing. Okay, so I'm going to switch gears now. Because I want to talk about, because I actually did talk about in my title something about resilience. So I want to talk about resilience. And my favorite study of resilience is the religious orders study, which is now known as the Nun Study. Some of you may already know about this because I think I've talked about it in previous lectures. So if you've heard about it, I'm just reinforcing that message. I tell my students that repetition is the mother of memory. So if this is the second or third or fifth time that you've heard it, that's fine by me. So the Nun Study is a longitudinal study of aging and Alzheimer's disease that started quite some time ago in the USA. And here's the book that was published by the main author, the main researcher, David Snowden, Aging with Grace. So the participants were 678 members of the School Sisters of Notre Dame religious congregation, and they are now up to 75, anywhere from 75 to 106 years of age. So the researchers went and did all kinds of studies and measurements and tests and so forth, but they also went back to the essays that the sisters wrote to gain entry into the society. And they were around, of course, about 22. So it turns out that the essays, if there was a lack of linguistic, what they call density, complexity, vivacity, fluency, the use of extensive phrases and adjectives and so forth, that functioned as a significant predictor of its author's risk for developing Alzheimer's disease later in life. So roughly 80% of the nuns whose writing was measured and was recorded as lacking in linguistic density went on to develop Alzheimer's disease in older age. Meanwhile, those whose writing didn't was actually really, really good. Only 10% developed the disease. Unfortunately, I can't go back to my childhood and learn to write better. So that's kind of the downside of this kind of thing. But it's kind of interesting that already earlier in life, there's some predictors of risk, anyway, for developing Alzheimer's disease. But the good news is, but there's also good news that has received much less publicity out of this study. So, many of the, and of course, the wonderful thing about this study, first of all, is that there's a very consistent kind of cohort of people who had very similar life experiences once they entered the sisterhood, right? So, and they weren't drinkers, supposedly. I'm going to take that as a matter of faith, but relatively healthy lifestyles and so forth. Anyway, so many of the nuns that didn't show AD, when they eventually passed away, and they had kindly donated their brains for study afterwards, many of them showed pathology characteristic of Alzheimer's disease, and yet they weren't showing the disease symptoms. So, I find that quite inspiring because it shows that somehow, for some reason, they have resilience. The brain was able to kind of be resilient against the onslaught of the pathology. So they kind of had the disease, but not the symptoms. Okay, maybe if they lived longer, eventually, it would come, right? But they lived a full life, had a good quality of life for long enough. Eventually they pass away for some reason, but they aren't having that dementia as a result of the disease. And somehow they were resistant. So I think this is one of many types of this kind of work that shows that it's possible to have resilience for something like Alzheimer's disease, for example. So here's something similar. So here's a study some years ago, and now there have been a number of studies like this, particularly in Western society, although not in New Zealand. But so here's a study in the UK, where they studied two 8,000 person cohorts of people who were 65 years and older. And one of the cohorts, they were studied over a 5-year period from 89 to 94. And another one, so the same age band, but now they just come half a generation later, or maybe it's a generation, 2008 to 2011. Okay? So they compared the rates of Alzheimer's disease in these two cohorts. So based on the rate of Alzheimer's disease or dementia in the first cohort, they predicted there should be 8.3% in the second cohort, and it was only 6.5%. So this is a very common story now. And no one really knows the reason. But if we think back to the Nun Study, where the language ability of people early in life predicted risk for Alzheimer's disease, you can imagine that there's various things that might be contributing. A higher educational level, and people are generally getting more schooling now as the generations come through than they used to. There's better health care. There's better prevention of vascular disease. They probably are more aware of healthy lifestyle kind of advice, and because you can't open a magazine anymore without being told what a healthy lifestyle is. And so, in a way, society and people are self-medicating. They're treating themselves already. Of course, that also means they're living longer, so then eventually, the problems can still arise. But at least they're having a longer, high quality of life than earlier generations used to. And in part, that's because they are developing resilience against the disease processes. Okay, so how does the resilience come about? Well, we have various other cell types in the brain, which I mentioned nerve cells, but there's many other cell types called astrocytes and microglia, and there's an important barrier that separates the blood from the brain, so that things that pathogens, other things that are in the blood don't get so easily into the brain and so forth. So there's a lot of guardians and barriers that help keeping the brain healthy. And I just thought I'd throw this slide, which is a couple sections. The left-hand one is a section to the human brain with some of these Alzheimer's pathology called senile plaques. And you can see that the tissue is dotted with these senile plaques from an Alzheimer's disease patient. Some of them are kind of diffuse and some are more kind of aggregated, but you can imagine it's not a good thing. And here in one of our mouse models of Alzheimer's disease, we can show that not only are these red ones are the plaques in the mouse model, but all these little green dots around here are the microglia that have swarmed in defense to these plaques to try to eliminate them. Unfortunately, the plaques are too dense for these little cells to kind of chew up and swallow and get rid of. So they try as best they can, but they can't. And so that leads to an inflammation. So we end up with neuroinflammation from these immune cells that are being activated, but can't resolve the problem, so they remain continuously activated. So we have continuous inflammation in the brain, and that in itself drives some pathology and loss of memory mechanisms and so forth, just from the inflammation alone. So inflammation is one of the things, in addition to the actual cell death and all the rest of it that's going on, that's important. So what we know is that people with, this is a sort of diagram of showing that people with more, what we now call cognitive reserve, the ability to maintain cognition in the face of pathology, they can function for longer. So people with low reserve might follow this course. With memory test scores, they fall off more rapidly over time and with the disease state than if you have in a more resilient cognitive reserve state. And eventually it falls off, okay? So the disease is kind of implacable eventually. But if you've lived long enough and enjoyed a much longer quality of life, then it's to your benefit, of course. So how do you build cognitive reserve? It's like, how do you build muscles? You got to use it or lose it. And it's not about having more muscles. When you exercise, you don't get more muscles, right? But you make them stronger and more, hopefully more flexible, and more able to do what you want. And the same is true for the brain. You need to use it or lose it, basically. And when you're using it, you basically are not adding to the number of neurons, although that small amount of neurogenesis I spoke about before does carry on and improve. But you're changing the connections between the brain. The more connections, the more the cells are talking to each other, by and large, the better. Even if you lose a few cells, the ones that are still there can still be good and functional. So here's just a few of the things that we all know that are good for you. You should stop smoking. You should exercise. You should adhere to a good diet. You need to be socially engaged. People have kind of underestimated that over the years, but now it's become very clear that social engagement, isolation leads to depression, leads to other things that are kind of aggravate the risk factors. So social engagement is good, controlling cardiovascular disease and diabetes, hypertension, all these things are part of building the reserve of the brain to withstand pathology. So basically, I need to move on. So I'm waxing lyrical here, but I want to get to this one. There are many proteins that the brain naturally has that helps its normal function and helps keep it kind of ticking along. And the one protein that we're quite interested in is one called secreted APP alpha, and I'm going to call this SAPA. And we have a lot of interest in this because, like, for example, here's a study where we show that it's neuroprotective. So in this little bit of the hippocampus that we have here, red means dying cells. So here we have a tissue culture of the hippocampus, and there's a few dying cells. But if we add a toxic molecule called NMDA, don't need to know what it does, means, but you can see that it kills off these cells very readily. There's a lot more red cells that are dead or dying. But if we give this neuroprotective protein, secreted APP alpha, into this tissue culture, the ability of this toxic molecule to kill off nerve cells is much reduced. So it's a nice neuroprotective molecule. So how can we harness this? So with colleagues, Stephanie Hughes in the biochemistry department, we've been working to develop a gene therapy strategy to try to harness this secreted APP gene and protein so that it can be used to treat our mouse models of Alzheimer's disease. So how does this work? Because it sounds kind of, because basically gene therapy involves viruses. Now you might think, that doesn't sound very good. Why would you use viruses for gene therapy? But here's the thing. A virus has infectious genes in it. And so, this envelope allows this virus to get inside cells. So what we do with gene therapy is we get rid of all the virus genes, so it's no longer infectious, and we put into the virus envelope the gene that we want to have inside cells. Okay? So we're using the virus as a Trojan horse. Okay? We're going to allow it to get inside cells and deliver the gene that we want these cells to have more of. So we want them to have more of our SAPA gene. So Stephanie's lab packages it into virus. We then inject it into, in this case, mouse brain, into the hippocampus, our favorite structure we're trying to rescue. let the cells start producing more and more SAPA, which it already does to some extent, but it's probably not as much as it should be in these Alzheimer's mice, who are expressing genes that we know in humans causes Alzheimer's disease. So these mice get plaques and so forth as well. And then we wait a few months or weeks or months and we see what happens. So if we come back to this long-term potentiation result, which I showed you before, we have our recordings and then we give our high-frequency stimulation, very brief, to cause our long-term potentiation effect like this. In the Alzheimer's mice, the long-term potentiation effect is not as good. So they're impaired in their ability to have this kind of memory mechanism activated. But our mice who had the gene therapy with the SAPA protein were rescued back to normal mouse levels of memory or long-term potentiation. And Valerie Tan, who was my PhD student at the time, and Stephanie's was the one who mostly responsible for doing this work. We also did a memory test with these animals. They get put in a pool of water with a hidden platform. They can't see and have to swim around and try to find this platform. And then once they find it, then they should remember where it is based on the kind of cues around the room and so forth. And so over many trials, they learn they take a faster and faster and shorter approach to find the platform normally over time. But our Alzheimer's mice have difficulty with spatial navigation. And you may note that many people with Alzheimer's disease have the same problem. Right? Spatial navigation is one of the things that goes reasonably early and can be quite severe. People can go wandering off and forget where they're going. Our Alzheimer's mice, who had the gene therapy with SAPA, were getting back towards normal learning performance on this task. So we're quite excited by this possibility, and we're doing lots more studies now to try to follow up on this kind of work. But here's the interesting thing. I showed you before a picture with red dots from a mouse brains that have these plaques. So here's a normal mouse, no plaques whatsoever. Mice don't get Alzheimer's disease. That's the wonderful thing about being a mouse, right? You can live as long as you like, and they never get Alzheimer's disease. So that's why we have to have these kind of animals where we have them expressing human genes that we know that are important for Alzheimer's disease. Otherwise, we'd never be able to do these tests. And I hope you understand that these are tests that we can't just do in humans. We have to do some animal testing first in order to understand how these things work and whether there's even hope of making these into therapies. So here's our example from our Alzheimer's mice. We got studded with these plaques that are there. And our gene therapy had made no difference whatsoever to the plaques. So that still had the pathology, but the brain was more resilient and cognitive function and many mechanisms were restored nonetheless. So we have not cured the disease, even for these animals, right? But we have created a situation where we've made the brain more resilient. And I think in many respects, this is the way the field is going to go. Of course, we're going to try to find ways to get rid of the plaques and so forth. That's been an abject failure in all clinical trials. And people will still be working on this as well we should. But I think the things that we do ourselves, exercise, diet, medical care, gene therapy, are options for keeping us going even if we have some pathology. And this is not just about Alzheimer's disease. This has potential for head injury, for stroke recovery. This protein it's not just about a particular disease. It's about making the brain resilient. Okay, so this is the take-home message. Healthy brain, healthy productive living during aging. This is what we're about. This is what Brain Health Research Centre in Otago and the Brain Research New Zealand across the nation that I am co-director for is very interested in. And of course, this is the sort of thing the Neurological Foundation is very interested in as well. And just remember, what is good for your heart is good for your brain. And with that, I'd like to close. I have a very fine lab group, some of whom are here tonight. I need to thank them because really I just sit in my office all day and just twiddle my thumbs. But they are the ones in the lab doing all the hard work, so I really need to thank them. And I need to thank all the funding agencies who get funding support from your tax dollars that come from the government and through the Health Research Council and through the Marsden Fund, but also through people's 30 years or however many years of kind donations to the Neurological Foundation has helped support us as well. So thanks very much for your attention.